Today we're going to talk about RNA expression analysis part 2, which is going to largely deal with RNA sequencing. So RNA sequencing, or RNA-seq, is a massively parallel EST sequencing technology. EST sequencing started in the early 90s and was basically sequencing of random cDNAs. And while it was great for gene identification, it was not ideal for quantification. It was expensive to generate the data, and the data quality were relatively poor, and there was a lot of non-uniform depth of coverage. For example, while one library may have 1,000 cDNAs, EST tags available, another library may have 10,000, which made differential gene expression tricky. Massively parallel sequencing, which sort of over the last 5 or so years has become de rigueur, overcame many of these problems. In fact, today, 50 million reads per sample in a 100 base pair paired end Illumina run costs about the same as it does to run an array. However, the sequences are usually shorter than EST sequencing tags, which today go about 800 base pairs. So they're not quite as powerful for looking at splicing or for looking at splicing. So here's just a really brief schematic on how RNA-seq works on the Illumina platform. First, you take an mRNA, and you can either fragment the mRNA prior to labeling, or you can reverse transcribe and then fragment the fragment cDNA. In any event, once you have the fragmented reverse transcribed cDNA, you would next adapt those sequences with small adapters and sequence them on a high-throughput sequencer, for example, Illumina HiSeq. After the data is generated, these sequences are then mapped to the genome, and you generate intensity values on an exon by exon or gene by gene or splice junction by splice junction basis. So Illumina isn't the only technology to do RNA sequencing. 454 sequencing, or pyro sequencing, is also quite effective. The read lengths are longer than Illumina sequencing. They can go up to 400 base pair, but in general, the number of reads is much lower, and so the cost per for generating enough data to actually do a differential gene experiment is quite high. Likewise, the ABI solid platform can generate data. The read lengths tend to be shorter, and to generate the data isn't quite as inexpensive as it is on the Illumina platform. So today we're going to talk mostly about the Illumina platform rather than the other platforms. The first aspect that is sort of a tricky one with RNA sequencing is the mapping problem. So once you generate the data, mapping, especially of transcript data, is actually quite tricky. And that's because transcripts are spliced, and gapped alignments are much harder than ungapped alignments. They're computationally more intensive. They take longer. Confounding this is the observation that most of the read lengths that we're generating with RNA sequencing are relatively short compared to EST sequences. Also, error rates of the high-throughput sequencing are relatively high when compared to Sanger methods, and highly biased to the 5' and 3' ends. Splice donor and acceptor sites, when you're mapping, often aren't clear, so you can have mismapping occurrences. And cDNAs often have repetitive sequences. There's a number of ALU repeats that are in thousands of human cDNAs, and these repetitive sequences can be extremely difficult to map accurately, if not impossible, without a paired end. There are many available algorithms to sort of tackle this mapping problem. Some of the most popular are TopHat, GSnap, MapSplice, and RUM. And it's safe to say that none of them can do the job perfectly, and all of them have their quirks. So just an illustration of mapping a spliced RNA. Here we have a pre-mRNA sequence which contains exonal sequences in orange and introns indicated in black. And when the pre-mRNA is spliced, obviously it makes a contiguous mRNA sequence. And short reads can map either within an exon, entirely within an exon, or between exons and over an intronal junction. And the reason that mapping this computationally can be tricky is these introns here, these black sequences, can be tens of or even hundreds of kb long, which makes the mapping problem quite difficult. After the mapping step, you try to arrive at a measure of the intensity of the expression. And this can be done, as I mentioned earlier, at the gene level, at the exon level, or at the splice junction level. And at the gene level, the method that appears to have gained the most traction is the RPKM method, or reads per kilobase mapped, millions mapped, reads per kilobase per millions reads mapped method by Mortazavi et al. From the Wold lab. And the advantage of this method is it normalizes both for the cDNA length, so the probability that you'll have more sequence tags mapped to a longer cDNA is higher than for a shorter cDNA, and also sequencing depth. So you might have one sample that's sequenced at 20 million read depth and another one that's sequenced at 30 million read depth, and this RPKM method gets around both of these limitations. However, depending on the sampling, for example, 10 million versus 100 million reads, the underlying distributions of gene expression can be quite different between the two, and that really complicates differential expression analysis. So this is an area that's under rapid development, and new methods are being generated every day. You can also do quantification at the exon level. There, typically, you could compute on exon junctions, sort of a variant on the RPKM method. But this is also largely an unsolved problem, and there is no perfect solution. So I think in this particular arena, the quantification arena, there's a lot of room for bioinformatic improvement. Because of the emitting the intensity values is tricky. Determining differential gene expression is likewise tricky because it depends on that step. So in addition to biological noise, a la the array studies we talked about in the last lecture, the number of reads per sample can vary considerably, even when doing similar samples in a multiplexing environment. So even though you think you've done everything exactly equivalently, you could still have one sample sequenced at 30 million read depth and another at 75 million read depth, and that can complicate differential expression. In addition, compared to array studies, the molecular biology for RNA sequencing is a lot more variant. So there are protocols that bias sequence towards the 5' end, strand-specific protocols, for example, towards the 3' end, polyase protocols, for example, small RNA protocols, for example. And all of these different protocols can make comparing data generated between them very tricky, if not unadvisable altogether. And obviously, mapping errors can have a major impact on differential expression. That prompts the question, how deep do you actually need to go? So here on this graph, we've taken mouse liver and sequenced it at up to 90 million reads of 100 base pair paired end sequence. And we've taken the data and mapped it across all exons from RefSeq, and then looked at the number of exons that are covered by one or more read, by more than 2 reads, or by more than 5 reads. And when you look at this, what you can see is that there's a rapid increase in the number of sequences mapped as you increase the read depth, and then you sort of have this plateauing function towards the end. So if we take the most conservative condition, which is more than 5 reads per exon, here you can see that we've largely plateaued at about 50% of transcripts, which compares pretty favorably to DNA arrays, where 50% expression would be on the high side. And if you went out another 100 or 200 million more reads, this value would continue to rise. And so when you talk about trying to determine how deep you need to go, you need to consider what your ultimate goal is. If it's every last transcript, then more is better. 200 million reads, you will still probably find new transcript expressed in your sample. However, if you're interested more in if you're interested more in splicing, more coverage is better. The more reads you have across a junction, the more confident you can be in determining that event A occurred and B quantifying the event. Also, different types of sequencing require different amounts of coverage. So total RNA sequencing requires more sequence than poly-A sequencing. And of course, as a lab head or a PI or a graduate student or postdoc doing the work, cost could very well be a factor. As you could see on the previous graph, there's a plateau and diminishing returns. So you have to decide whether or not doubling the cost by generating twice as many reads is worth a 5% incremental improvement in detecting gene expression. If we compare methodologies in RNA sequencing with that from DNA arrays, which we talked about in our last lecture, you can see actually there's many similarities and many differences. Although the principles are quite different, one is hybridization based and one is cloning and sequencing based, they have commonalities and differences. So with arrays, a big difference is you actually do need a genome before you can design the array. And in fact, if it's a cDNA, you actually need the clones at handy before you can actually make the array. With RNA sequencing, you don't practically need a genome to do it. And I'll show you an example later of denovo RNA sequencing. But if you're interested in really going in deep into a transcriptome and doing differential gene expression, it really does help to have a genome and an annotated genome at that. Likewise, while cross-hybridization is a problem for DNA arrays, theoretically it's not a problem for RNA sequencing, but practically when you're mapping sequences, especially repetitive sequences, it does become an issue. It's not exactly cross-hybridization, but you can have a sequence tag mapped to more than one location in the genome, sometimes more than one transcript in the genome. With RNA sequencing, because you have base level resolution, allele specificity is possible, and in general, that's not the case for DNA arrays. Although, depending on how you design the arrays, it can be possible. Likewise, splice forms can be detected by both arrays and by RNA sequencing. However, with arrays, you need to actually tile down either the junctions or the possible junctions. Whereas with RNA sequencing, you don't need to know the splice forms de novo. Again, with arrays, you really can't detect new genes entities that you have not put down on the array, whereas sequencing doesn't require you to know what's there a priori. And both technologies can accomplish strandedness. So arrays, you could put down either strand if you need to, for example, with short oligonucleotides or long oligonucleotides. The dynamic range is better for sequencing depending on read depth. If you sequence 50 million or 100 million reads on a sample from complex vertebrate genome, you have thousands of fold of dynamic range. Likewise, if both technologies are highly reproducible, and I'll give a slide in a minute that will demonstrate reproducibility of both technologies. And cost-wise, to generate the data, they're approximately the same depending on how you generate the data. But with RNA sequencing, there are added computational costs which are not insignificant and in fact make up the larger challenge in analyzing the data. So our first question, what are the differences between DNA arrays and RNA sequencing for gene expression? A, RNA-seq can have better dynamic range. B, RNA-seq can be stranded. C, arrays are easier to analyze. D, A and C, or E, A, B, and C. And the answer is A and C. RNA-seq can have a better dynamic range. I say can because it depends on the read depth. And C, DNA arrays are generally easier to analyze. So let's talk about whether or not the sequences are comparable. So we're gonna do an experiment here where we're gonna do a head-to-head comparison with exactly the same samples. Our samples are going to come from a C57 black 6 mice, from 3 to 5 mice per sample, taken at 3 different times of the day on 2 consecutive days. So we have pooling, so we have overcome biological error and independent replicates. So we have biological error and technical error addressed there. The arrays were done on the Affymetrix platform, and RNA-seq was done on Illumina. And we looked at how these replicates compared both between and within technologies. So here are the comparisons. On the top of the slide, we have RNA sequencing, and this is replicate 1 here and replicate 2 here. And if you look at all the genes that are expressed in this particular tissue, liver, and you do an XY plot, you can see that there's extremely good reproducibility between sequencing, an R-squared value of 0.98. And this is at one time of the day. There's 2 other cases, and it's nearly identical. Again, extremely reproducible technology. We took the same samples and put them on exon arrays on the Affymetrix platform. And again, with replicate 1 and replicate 2, you can see very good reproducibility, maybe even slightly higher than RNA sequencing, and an average R-squared value of 0.98, although I'd want to do that experiment several more times before I concluded that both technologies are extremely reproducible. However, when we look across or between technologies, plotting the values from arrays on one axis and the values from sequencing on the other axis on a gene-by-gene basis, you can see that the R-squared value isn't quite as good. And that's because of the of both dynamic range issues, but also other factors. So what reasons could explain this? A, different dynamic ranges. B, RNA-seq looks at one region of a transcript, whereas arrays look at more than one region of a transcript. C, quantification by arrays isn't as good. Or D, arrays require the gene to be annotated. And here the answer is A. The 2 technologies have different dynamic ranges. It's true that RNA-seq can look at one region of a transcript, but in general, you're sequencing the whole transcript. You're not doing 3' biased. In general, you're sequencing the whole transcript. Quantification by arrays we just showed was just as good as RNA sequencing. And it's true that arrays require the gene to be annotated, but that doesn't have anything to do with whether or not the 2 technologies don't compare. So, of course, the real reason that most people do RNA sequencing instead of arrays is to get at alternative splicing issues. And this is just a really brief overview of alternative splicing events, which can include constitutive splicing, where you have the same splice form made every single time, exon skipping events, where you could have one exon, in this case this one, skipped in the mature transcript over here. You can have intron retention, in which case a small chunk of the intron is retained and in the mature transcript. You can have mutually exclusive exons, where you have 2 very different transcripts that are made. And of course, you can have alternative 5' and 3' splice sites. So different forms are made because of the alternative forms. And in RNA sequencing, you can find all of these events. So here's an example of looking at the mapping out splice junctions for one particular gene, SYT1. And what you can see is that the various exons, which are here at the bottom of the screen, have reads that span, multiple reads that span various exons. So here you can see reads that span between this exon here and this exon here, and pretty much all of the exons in the transcript are covered by these events. And from looking at these events, you can find issues where you have retained introns, alternative exons, mutually exclusive exons sometimes, and the various other events. Another application of RNA sequencing can be disambiguation of overlapping transcripts. So here's an example of 2 transcripts, NR1D1, which is reverb alpha, and THRA, thyroid hormone receptor alpha. And what you can see is that they both have a similar expression profile over the day in the mouse liver. And it so happens that these transcripts are head to tail in the genome, so that one goes like this and the other one comes back, and they have an overlapping region in their transcription. And this happens to be near where probes are often designed with arrays to detect their expression. So it was a little bit difficult to figure out what's going on at this particular locus simply looking at array data. However, although the messages overlap, in this case, because we have paired end sequencing, we can use a read upstream or downstream to be able to find out which transcript this particular read matches to, either NR1D1 or THRA. So in this case, we could tell that most of the reads at this particular locus are mapping unambiguously towards NR1D1 and not towards THRA, and therefore the probability is that our array signal is artifactual. Another example of the power of RNA sequencing is to detect new entities, in this case looking at microRNA expression. So this is a cluster of microRNAs that are made off the same transcript, and when we looked at this particular locus, we were able to see that they had likewise a variation in their transcription over the day. So here you can see this peaks here, here, and here. And in this particular case, when we quantified these regions, we found that all of these transcripts, which are the mere 292 cluster, basically had this same identical pattern, and the parental transcript does as well. One other sort of interesting thing that you can see with with sequencing data that you don't get with arrays are regions of the genome that you didn't query. In this case, we're looking at a particular gene, D-box binding protein, or DBP, and DBP has this exonal structure. Here's exon 1, exon 2, exon 3, and exon 4. And what we're seeing here in our RNA sequencing data is actually quite a bit of signal here between exons 1 and exons 2. And when we look at the expression of the spliced form of this message, we see a robust oscillation in the transcription, and we see the similar robust oscillation in the transcription of the intronal sequence, so a read here and then a read downstream. And those reads are also quantified not just by RT-PCR but also by northern blots, where you can see the conservation of this expressed intronic sequence. One of the most interesting developments over the last decade or so has been the discovery of non-coding transcripts that have functional consequences. And these are sometimes called links for long non-coding RNAs. And so here are a couple of links, one on chromosome 6 and one on chromosome 7, where from denovo, from looking at RNA sequencing, and we found novel links that haven't been annotated previously that have this variance to their transcription over the day. So it's another type of information that you get from RNA sequencing that you don't get from arrays typically. We talked earlier about one of the advantages of RNA sequencing being de novo RNA-seq. And so here's an example of de novo RNA-seq. In this particular case, we wanted to sequence our favorite genes from Syrian hamsters, which don't yet have a genome sequence. By the time you read this, it probably will have a genome sequence. Genomes have been dropping like flies, no pun intended, but today they don't have a genome sequence. And so RNA-seq can do this while arrays can't. And so what we did was sequenced poly-A plus RNA from liver from the Syrian hamster and 165 million reads. We used an algorithm called Trinity from Aviv Regev's group to do de novo alignments of this de novo RNA sequencing, and then we took those assemblies and did BLAST-X to find species orthologs. And in this particular case, we were able to find 16 of the 18 factors that we were interested in. So I'll show you an example. Here's a gene ARNTL or BMAL1. The mouse form is in red on top, and that's the annotated full-length form of the gene. And from mapping our hamster assemblies onto the mouse form by BLAST-X, we were able to find we're able to find a big chunk of the hamster form. So in this particular case, the assay worked pretty well. So here we have 2,884 reads, or approximately 100x coverage of the transcript, and we're still not getting all of it. And we'll talk about reasons for that in a minute. Here's an example where it worked okay, but not quite as well. So here's an example of a gene called AHR-GAP17 that doesn't exactly roll off the tongue. And here's the hamster assembly that we got from Trinity. And when we compare that to the mouse form, we actually see this pretty weird inversion where the mouse form, which of course normally goes 5' to 3' and works fine. In this case, because of the eccentricities of the algorithm, it actually mapped the hamster form incorrectly. So while the hamster form should go like this and like this, it doesn't actually go that direction. So it's gotten mixed up. And this could be because the sequences here are repetitive, and it's just simply found two matching sequences, or for just strange reasons. So despite the 100x coverage, the cDNA appears incomplete for ARNTL or BMAL1. What could explain that? Is it A, poor representation of the 5' and 3' ends of the cDNA due to library construction methods? B, mapping issues for repetitive sequences? C, species differences? D, tissue-specific transcripts? Or E, all of the above? In this particular case, it's all of the above. We could very well have poor representation of the 5' and 3' ends. One of the things that happens with sequencing is there's a fragmentation step, and so frequently the 5' ends of a gene have the sort of step function, and the 3' ends of the gene also have a step function, and this is largely due to the sonication step in fragmenting the gene. It could also be due to repetitive sequences. We've talked about that a little bit. They can be very difficult to map, and therefore we may have excluded them in the de novo mapping. There could be species differences. So the 5' end of the hamster gene could be very different from the 5' end of the mouse gene. There could also be tissue-specific transcripts. So remember, most of the time we're looking at a ref-seq transcript, a reference transcript. And that reference transcript may have come from brain or from liver or from adrenal gland or what have you. And the form that we're looking at in this particular tissue may be different. And so all of these answers are correct. Another application of RNA-seq is going after base level changes, which can occur during things like allele-specific expression or during RNA editing. So base level expression allows for detection of both of these events. And there's an example of allele-specific expression analysis using RNA-seq in the Gregg et al. Paper from Catherine Dulac's lab, which was published in Science in 2010. And there's an example of A to I editing events from Jinbilly Lee, who's now at Stanford, when he was in George Church's laboratory in Science in 2009. These methods rely on deep, accurate coverage of actually RNA and sometimes DNA too, when you need to measure the DNA of, for example, an outbred population like humans. And this can be technically challenging. So here's a case of looking at allele-specific expression. If you want to think about it in a simple, straightforward fashion, in this case, we're going to set up two reciprocal crosses. We're going to have one strain of mouse, for example, a C57 black 6 strain in blue, or an outbred strain, Castaneous strain, here in red. And for a gene that has classical biallelic expression, you expect that each allele is going to be equivalently expressed when you cross these two animals. However, for a paternally expressed gene, you're going to see expression of only the paternal allele and the exclusion of the maternal allele. And for a maternally expressed gene, these two crosses down here, you see the expression of only the maternal allele. And this can happen during processes like imprinting. So here's an example of what that looks like at the base level. So here's two conditions In the top, we have a biallelic expression condition. In this case, we're going to look at base level changes. So we're going to measure the paternal and the maternal alleles separately and by themselves. And here we see that the paternal allele has an A at this position while the maternal allele has a T. And from a biallelic expressed message, we're going to see roughly the same number of both alleles in the F1. However, in an allelic imbalance, when you cross these two animals, here we're going to see overrepresentation of the paternal allele. There's more A's here than there are T's here, and so we're going to call that allele-specific expression. In this case, it's relatively dramatic, but not a complete imprinting event where 100% of the transcript could come from the paternal allele. So what are the confounding factors in determining allele-specific expression? Is it A, DNA differences, B, alignment errors, C, depth of coverage, D, poor adapter tripping, or E, all of the above, or F, B, C, and D? And in this case, the answer is F, B, C, and D. So alignment errors can frequently introduce what looks to be a mutation, but are actually more alignment errors than a mutation. It's also extremely sensitive to depth of coverage. If you have very poor depth of coverage, your ability to detect an allelic difference is pretty poor. Because of the relatively short read nature, a lot of the sequences that are generated from these machines are adapters, and if you have a failure of adapter trimming, parts of the adapter can look like the native locus, and you can have what looks to be a mutation when it's in fact not a mutation but part of the adapter. And the reason I'm saying that they're not DNA differences isn't that there aren't DNA differences between various strains, but because you've measured both the paternal strain and the maternal strain, you know that the allelic differences aren't due to detection differences, but they're rather due to expression differences. So I've talked today mostly about poly-A-seq and a little bit about total RNA-seq. So there's obviously other versions. So like I said, total RNA-seq is very much like poly-A sequencing, but preserves messages that don't have a poly-A signal. For example, like the links that I showed you earlier. In this case, ribosomal subtraction is required because ribosomal RNAs make up about 99% of RNA in a cell. And so this step is a critical step for doing total RNA-seq. There are many small RNA sequencing protocols now to get at these classes of small RNAs, like microRNAs, that are in samples. In this case, the RNA can be purified while preserving small RNAs that are lost in some column and gel preparations used for doing poly-A or total RNA sequencing. And this technique is used to define new classes of small RNAs or measure their differential gene expression. There's also ribosequencing, where you use antibodies to precipitate RNA associated with ribosomes. These are then labeled and sequenced, and this is thought to be a more representative of what's going on translationally in a particular sample. And virtually every month or so, there's a new form of sequences, including 3P-seq. So just to finish up, we're really at only a few years after the original description of sort of the modern RNA sequencing technologies. So the technology is still in its infancy. A lot of the improvements are needed, for example, in determining expression values on a per gene, per exon, per allele, per splice variant form. This is still very much an unsolved science. Many improvements in molecular biology and informatics means that these technologies will eventually replace arrays. We're not there yet for all applications. Arrays still have some legs left in them, but pretty soon the RNA-seq will supplant DNA arrays. A big advantage is that you don't really need an a priori transcriptome, but again, that depends on what your actual needs are. If you're just looking to find a particular gene, as in the example I showed you, denovo RNA-seq can work pretty well. If you want every last base pair of every last gene, then denovo RNA-seq is not a perfect technology. There are many challenges in RNA sequencing. The molecular biology is tricky. All sequences don't amplify equally, so you have a bias that's inserted by your molecular biology. And the biggest challenge here is really informatics. Storing and manipulating this data is extremely challenging. I mentioned several of the issues, including looking at the expression levels, mapping the data, and these are still a very rapidly changing and rapidly improving scenario, but still very far to go. And that's our lecture.